select the best 5% for their presentation. We will, in the same manner, we will start the presentation. And which are going to be the best among the first category, that is paper presentation on the basis of hypothesis, that will get awarded first, second, and third platinum, gold, and silver. Likewise, outcome-based platinum, gold, and silver awards. Likewise, the other category, that is the category of poster presentation, again hypothesis-based and on the basis of outcome-based, 3 awards, platinum, gold, and silver, respectively, in both the groups. And in the startup also, 3 awards are there, that is platinum, gold, and silver. To take more and more participation so that you will get the more and more exposure. And we must thankful to the SARS novel coronavirus. If such pandemic was not there or is not there, we are not at all in a position to join each other on this platform. This is the positive side of this COVID-19 pandemic. We all are staying at our home and we are just utilizing that time in this manner. So, as I always said and saying also, take more and more vitamin P as you can, so that more and more positivity is with you.
Thank you so much, ma'am. And I'm extremely sorry to interrupt you. I hope that all the participants are joined in this meeting now. If anybody is left, kindly just give their email ID in the chat box so that we can join them. Thank you so much.
So shall we start?
I think so, ma'am. Yes, most of the participants have joined. I think so.
Okay.
Yeah.
Now we are going to start. Welcome, dear participants, and join us with a happy heart. Giving encouragement to others is a most welcome gift. For the result of it are lifted spirits, increased self-worth, and a hopeful future. So, you all are very welcome to the 5th workshop, that is how computer helps in the designing of biological data under Consortium Biotech workshop. It must appear in other ways than words. As everybody knows very well that this Consortium Biotech workshop is a series of 5 workshops. We have already completed the first 4. Just how to start the lab work under aseptic conditions, how microbes are fascinating, how the beauty of acids, that is in the form of DNA and RNA, how the gene is expressed in the form of protein. Now, we are just towards the end of this Consortium Biotech workshop, that is workshop 5, how computer helps in the designing of biological data. This workshop is, as usual, divided in the 4 sessions in 2 days. The first session, that information retrieval from different databases. Then second session comprises of FASTA, BLAST. These are the 3 sessions which we are going to deal today. And from tomorrow, in the 3rd and 4th session, that is on tomorrow, first in the 3rd session, genome map analysis, followed by primer designing, protein designing, and protein-protein interaction, followed by the feedback session. When we are saying workshop, here we have just pointed out in the digital manner, in which manner we have to just improve our practical skills or laboratory skills. Here, our panelists will just do each and everything and show you how to do the BLAST, how to read out the genome map and how it will be analyzed, How the primers are designed with the help of various softwares, likewise the protein designing and protein-protein interaction, which tools are required, which methods are useful for such designing. When we are talking about again and again biological data, designing of biological data, how computer helps in to do so. Whenever we are even planning of any of the experiments related with the biological data, it means there is a requirement of some concrete data which is useful for the designing of our experiments. Either that experiments are related with the routine labs, or we can say that experiments are related with the designing of undergraduate, postgraduate, doctoral or postdoctoral fellows work, or that are related with our own project works. Why is it so? Whether we are going to copying it? No, not at all. Whatever the work already has been done, that is the groundwork of the proposed work. That's why there is a requirement of some data, some results, some literature which is helpful for us to design our new and the novel work. So, the first session of today's workshop, how computer helps in the designing of biological data, is mainly focusing on information retrieval from different databases. When we are using the term database, what is this database? This mainly a collection of structural information searchable in the form of table of contents. That is, we can say that index, or that index, that structural information which is searchable in nature, is mainly periodically updated in the form of release of the new editions. Once it is periodically updated in the form of the release of the new editions, that should have cross-reference. That cross-referenced are in the form of hyperlinks. That links are linked with the other databases. Hence, we are considering this, all these features which are present in the database that consists of basic unit, which is known as records. Each records consists of fields that is considered as predefined data related to the record. Suppose we have taken an example, protein databases would have protein entries as records and the protein properties as fields. Which type of protein properties? These are name of the protein, protein length, amino acid sequences, etc., etc. Hence, that data should also have associated tools in the form of softwares. Why there is a requirement of the softwares? Why there is a requirement of the tools that are necessary for database access or query, database updating, or database information insertion or deletion. Therefore, there is a requirement of such databases which are useful for us. How can we say that whatever are the databases available, these are perfect databases? Any perfect database should be comprehensive in nature but easy to search, annotated but not too annotated, a simple, easy to understand structure. The most important thing is cross-referenced, minimum redundancy, along with easy retrieval of data. Hence, the purpose of biological databases are based on three important factors. That is, the first one is to disseminate biological data and information. to provide biological data in computer readable form. And the third one is to allow analysis of biological data. These are the three important purposes of any biological databases. Why there is a requirement of biological databases? N number of journals are there through which we are getting the information. N number of hard copies of these journals are there. N number of libraries are available almost in every institution. But that is not sufficient. That is not within the reach of us. What we are doing nowadays, we are not doing nothing. Only we are having a single laptop or computer along with net. And we all are connected with the help of this through this only two basic things, laptop or desktop along with the net. Whole world is inside whole world is inside our hands only because of these two. That's why, because exponential growth takes place in biological data that is very difficult to manage in any of the ways, in the form of excuse me, hard bounds, or in the form of hard copies, or in the form of soft copies also, but they are not in the managed way. That's why n number of researchers are going on in the field of biological sciences. That's why huge amount of data is generated. Which type of data is generated? That is in the form of genomic sequences, 3D structures, 2D gel analysis, MS analysis, microarrays, etc., etc. But these are no longer published in a conventional manner. Huge volumes of data are produced. How to manage these data? How to store these data so that others can also use whatever I have produced or anybody else has produced. Now, with the help of these databases, the researchers are having the facility to submit these databases, these data, whatever we have generated, to directly to the databases. And or in other words, we can say that this is the essential tool for biological research. These biological databases are the essential tool for the biological research. Initially, distribution of databases that was in the form of books, articles, computer tapes, floppy disks, CD-ROM, FTP, online services are also there. But once WWW, that is World Wide Web, was emerged out, was developed, was invented in the year 1993. From the 1993, when WWW was evolved, remarkable discoveries in the field of biological sciences, remarkable discoveries in the field of medical sciences, taxonomy, and so on, microbiology, each and every field related with the biology, only because of three W's. Hence, objectives of these biological databases are to recognize various data formats and know what their primary use are, to know, understand and utilize all types of sequence identifiers. Suppose we have just invented one organism, that is microorganism Pseudomonas citanaria, for the degradation of petroplastics. What we did? We sequenced it. And whatever the other factors, we are going to just discuss in the later slides. We have fulfilled the requirement, whatever is the requirement, to submit the sequence of that, partial sequence of that, or whole organism genome sequence of that to respective database. That's why to recognize various data formats and what their primary use. Another objective of the biological database is to know, understand, utilize all types of sequence identifiers. The third objective is to know and understand various feature types. All these are present in the GenBank flat files. If we are having all these in the GenBank flat files, it means to know and understand the various GenBank divisions also. Keeping all these objectives in our mind, how these databases are working. Suppose we are using the Google. Google is the most favorable one. This Google is acting as the search engine database. Through this search engine, display the results from database with the help of this Google search engine, which is having the web spiders. Just like the web of spider, web crawlers also, with the help of all these websites, www, World Wide Web, all these websites, they are crawls the websites, then copies the information in websites, store the information into database, and they are just displaying in front of us. In this manner, search engines work. Search engine, that is, this Google is acting as the search engine of the database. They are displaying the results from the database. How they are displaying the search engine? Because they are having the web, just like the web of the spiders, information web. They are just scrolling all the websites which are available. After crawling the websites, they are copying the information in the websites, storing the information into the database, and displaying in front of us. Once we are getting such type of informations, now more than 1,000 different biological databases are available nowadays, having the variable sizes from 1,000 kilobases to 10 gigabases. DNA is more than 10 gigabases. Proteins are 1 gigabases databases are available. 3D structure 5 GB databases are available. Others, either they are smaller, but they are available. Whatever is our need, either the smaller one or the larger one, everything is covered under all these databases. But these databases are also updated frequently on the basis of daily basis or annual basis as per the requirement of that particular database. Either they are daily on the daily basis or annually basis, their informations are updating frequently. How to find them? N number of links are available. Some representative ones are Amazon's, BioHunt, Google, etc., etc. Now, again, another question arises. What are the categories of databases for life sciences? Life sciences itself is a huge reservoir. All biological sciences, botany, zoology, microbiology, biochemistry, biosciences, genetics, human genetics, but and many more. These categories of databases for life sciences are sequences, that is sequences of DNA and protein, genomics, mutation or polymorphism, protein domain or protein family. They are having the specific tools. Proteomics, 2D gel, mass spectrometry, 3D structure, metabolism, bibliography, others in the form of microarrays, protein structures, protein designing, etc., etc. For when we are doing all these, again question arises, what are the ideal minimal content of a sequence database entry? Suppose I want to submit my sequences to a particular database. What are the minimal requirement? That Minimal requirements are sequences, accession number, which is in the form of unique identifier, taxonomic data, references. These are the four important things. What we have to give, what we have to provide to the database, just what sequences we want to submit. For that, there is a requirement of accession number, which is considered as unique identifier, taxonomic data, and references. On what ground you are saying these are the novel ones? These are not already available in the databases. For the annotation and curation, keywords, documentation. These are required for annotation and curation. Now we are having the databases, we are having the data, we are having all the information related with the biological system, which are available in the various databases. But how to access such distributed data? This biological data is widely distributed over the www, as it's in the picture from 1993. Then this data can be retrieved by either with the help of search engine or the data retrieval tools. As we just observed, Google is acting as the search engine. And along with that, n number of data retrieval tools are present. Once we are talking about the search engine, what are search engine? This is an information retrieval system designed to help find information stored on a computer system. These search engines are usually presented in a list and they are commonly called as hits. The most public visible form of search engine is a World Web search engine which searches for information on the World Wide Web. Most commonly used verb and search engine, that is World Wide Web, www. Some examples of these search engines are Google, Yahoo Search, Leapfish, Bing, Amazon, etc., etc. Why we have to use search engines? This can find relevant web pages, and it is also difficult if such engines are not available, search engines are not available. It is difficult to find desired information as well, and also difficult to find specific information related with our work or the desired one. These are some glimpses of Google database, Google, which is acting as the search engine, Leapfish, Bing, and Yahoo. How now after the data, that is search and engines, we are having the data retrieval tools. These data retrieval tools, they are dedicated to access information for molecular biologists. How to access? That is with the data, search engines are there, but how? That is with the help of data retrieval tools, which are mainly in the form of interest, DBJet, SRS. All of each of these allows text-based searching of a number of linked databases and sequence searching both. If we are searching for the text, they are allowing. If we are searching for the sequences, they are allowing, because these all are linked with the other databases also. Because these databases are just like the network, just like the web of the spiders. But they differ in the databases they cover. For instance, one database is covering PDB. Protein database is covering the protein sequences. GenBank, they are mainly focusing on the nucleotide, gene or gene function or other properties related with the gene. This interest is www Global Query Cross Database Search System is a federated search engine for data retrieval. This is developed by NCBI, National Center for Biotechnology Information. This entries is the primary text search and retrieval system of NCBI, largest and biggest system in the world, that integrates the PubMed database of biomedical literature with 38 other literature and molecular databases, including DNA and protein sequences, their structure, gene type, gene function, their gene expression as well. The largest database of the world. Interest developed by NCBI, worked on www. This covers the databases which are nucleic acid having the GenBank, RefSeq and PDB, protein sequences in the form of SwissProt and PIR. SwissProt, PROT. These four letters are the first four letters of protein, PROTEIN. From this prot is made. PROT is from protein, first four letters of protein. Whenever this is available with any of the words, that is SwissProt, immediately clicks in our mind. This is related with protein databases. Then 3D structure, MMDB, genomes. Many stores are there. Many sources are there to study the genomes. Along with this, PopSet from GenBank, OMIM from OMIM, taxonomy, NCBI taxonomy database, books, that is from the bookshelf, PropSet, that is GEO, Gene Expression Database Organization, then PubMed. All these are available on their respective databases. NCBI's largest search engine, which is considered as the life sciences search engine. Address the life sciences search engine. Whatever you want to search related with the life sciences or biological sciences, everything is available here. Whatever you want to search, just put here. Search across databases and go. Here also, search across databases. For instance, we want to just search insulin and go. Whatever are the data available related with the insulin, all will be within few seconds. All are available on our screen. PubMed is there. PubMed lateral is there. Site search is there. Books, OMIM, OMIA, that all are available. Nucleotide, EST, GSS, protein, genome, structure, taxonomy, SNP, and many more. This is the huge repository of all the fields related with biological sciences. Whatever you want, you just type here and go. Here, all bars are there on the basis of which, suppose you want to search from PubMed, you are going for literature, you just click here. If you want to go just search from all databases, click here. If you are just looking for the human genome, search here. Or you want any information, you want to retrieve from GenBank, we can go for this. That's why this is a collaboration of NIH, NIG, EMBL. These are the three major agencies, three major institutes, which mainly working on it, and they have developed the crystal clear, user-friendly system. NIH is with the help of NCBI, NIH with the help of NCBI, and NCBI is having the largest, that is interest for the submission and updates. Then they are submitting in the reservoir, that is GenBank, EML and PDB. Mainly they are focusing on GenBank. Then NIG with the help of CIB, that is Gentry, For the submissions and updates, they are mainly focusing on DDBJ, that is DNA Data Bank of Japan. And here EBI, that is through SRS. For the submissions and updates, then these are of EMBL and they are submitting to the EMBL. With this collaboration, they are working three main institutions, NIH, NIG and EMBL. They are working together for the submissions and updates. They're having their respective sites, but they are well connected with each other. They are passing the information to each other. When we are talking about such type of collaborations of these three institutions or these three agencies, when they are working together, then only these web is working in a systematic manner or systemic manner. Otherwise, what will happen? Sometimes some conjunctions are there, so we are not getting our desired information at that time. Sometimes you are getting the site is not working. Sometimes they not responding. These are the options when there is conjunction in such type of web. Then only such happens, otherwise not. Hence, over the past few decades, major advances in the field of molecular biology, coupled with advances in genomic technologies, have led to an explosive growth in biological information generated by the scientific community. We all are working in one way or the other in generating huge and huge amount of data. However, the data which we are generating, that is in the form of biological data, which are available in biological databases, which are considered as the libraries of the life sciences information, collected from scientific experiments, applications, high throughput experiments, high throughput experiment technology, and computational analysis. All these informations contained in biological databases, these include gene function, structure, localization, clinical effects of mutations, as well as similarities of biological sequences and structures. Hence, information on some popular databases which are available online for the gene sequences, for the protein sequences, to study the phylogenetic analysis, to just through the structures of various proteins, various pathways, and microarray databases as well. Such work started in around 1956, but once it comes in the picture, the word is bioinformatics. Bio, that is the biological system. Informatics, that is information from the biological system or information of the biological system. This is the application of information technology to store, organize, analyze the vast amount of biological data, which is mainly available in the form of sequences, structure of proteins, and nucleic acids. Once these all are available, all whatever are the data available in the various databases, they are storing there, organizing and analyzing this huge amount of data. Through this databases, there is a requirement of one controlling center. There is a requirement of one emerging branch that is known as bioinformatics. As its name suggests, this biological information of nucleic acids is available as sequences, while the data of proteins is available as sequence and structures. Biological information of nucleic acids is available only in the form of sequences. But if the data of protein is available, that is in the form of sequences as well as structures, either primary structure, secondary structure, tertiary structure or quaternary structure of any protein. Usually, these sequences are represented in the form of single linear chain, while the structure of proteins that contains the three-dimensional data of sequences. Hence, National Center for Biotechnology Information, that is NCBI, in 2001, they defines bioinformatics as bioinformatics is the field of science in which the merging of biology, computer sciences, and information technology into a single discipline. With the help of this bioinformatics, three different fields, that is biology, computer science, and informations, they are working together on a same platform. There are three important subdisciplines within bioinformatics. These are mainly based on number one, this subdiscipline, development of new algorithms and statistics. Why development of new algorithms and statistics? That is for the assess relationship among members of large data sets. The second subdiscipline of bioinformatics is the analysis and interpretation of various types of data, including nucleotides, amino acid sequences, protein domains, and protein structures. These are with the help of the development and implementation of tools that mainly enable such eminent access and management of different types of information. It means with the help of such type of data, we have to develop and implement of such tools so that they can be systematically, they can store, they can analyze, they can receive, and they can just provide the data at the time of need. The three subdisciplines within the bioinformatics, that is development of new algorithms and statistics, that was one subdiscipline. Another one is the analysis and interpretation of various types of data. And third, their implementation, that is with the help of various tools, just for the retrieval of the information. Racial database concept, that is based on the concepts of computer science and information retrieval concepts in the digital libraries, which are important for understanding biological databases. Concept of computer science and information retrieval concepts are very, very important to know which type of digital libraries are important for understanding biological databases. These biological databases, mainly for design, development, and long-term management, are our core areas of the discipline of bioinformatics. What are the three important things? Design, biological databases, how they have designed, in which manner they have to develop, and long-term management, how we have to store and manage each and every information in the systematic manner. This is the core areas of the discipline of bioinformatics. Data contents include gene sequences, textual descriptions, attributes, and ontology classification, citations, and tubular data. Hence, these are considered as or described as semi-structured data and can be represented as tables, key delimited records, and XML structures. Once they are considered as or described as semi-structured data, for what purpose? They are represented as tables, key delimited records, and XML structures. These are mainly present among the databases which are mainly using at the time of accession of databases on the basis of accession numbers. Now, once everything is managed in the systematic manner, that is, which type of database is available for which type of sequences, their submission, their store, their their analysis, their complete management. Now, we have to prepare such type of databases as we have just discussed. For that, preparation is for the collection of data. We have to organize in such a manner that that should be easily accessed, managed and updated. And all these collection of data mainly in the form of easy accessible manner, so that we can make it available to a multi-user system. At a time, same information can be gathered by n number of users at various places of the world. Hence, classification of databases is based on primary, secondary, and tertiary or composite databases. This primary database contains information of a sequence or structure alone. Primary, basic information that is related with the sequence or structure alone. Some examples of primary databases, these are SwissProt and PIR for protein sequences, GenBank and DDBJ for genome sequences, and the Protein Data Bank for protein structures. The second class of the database, that is secondary database. This mainly contains derived information from the primary databases. This approach widens biological knowledge and explores the great diversity of individuals. This is mainly based on secondary database is based on the information from the primary databases, but they are widens the approaches related with biological knowledge and explores the great diversity of individuals. This mainly contains the information like conserved sequences, signature sequence, and active site residues of the protein families, which all are arrived by multi-sequence alignment of a set of related proteins. This secondary structure database mainly contains entries of PDB, that is protein databases, which are organized in wonderful manner. These contains entries that are classified according to their structure, that is structure of the protein. What structure of the protein? That all alpha proteins, all beta proteins, turns, helix. These all are the parts of any structure of a protein. So whatever the entries which are mainly submitting here in the secondary databases, that are mainly focusing on the structure of the protein that contains all alpha helix, all alpha stress, that is all alpha proteins, all beta proteins, all turn helix. These also contain information on conserved secondary structure motifs of a particular protein. Some examples of the secondary databases, these are created and hosted by various researchers at their individual laboratories, which mainly includes SCOPE, that is developed at Cambridge University, CATH, developed at University College of London, PROSIT of Swiss Institute of Bioinformatics, eMotif at Stanford. The third category, that is third class of database, considered as composite or tertiary database, which is mainly focusing on tertiary, that is protein tertiary structure database. They are gathering the information particularly on the basis of experimental or predicted protein 3D structures. Which type of information is required for the 3D structures? Special positions of membrane-bound peptides, number one. Another requirement of this tertiary database is domain insertions, molecular structures, angens, other properties. These are required that all are related with the 3D structure of protein prediction, which we are obtaining from the experimental data. Which was the first database which was evolved? that was created within a short period after the insulin protein sequence was made available in 1956. Interestingly also, and incidentally also, insulin is the first protein to be sequenced. The sequence of insulin consisted of just 51 residues, which characterizes the sequence. And around mid 1960s, the first nucleic acid sequence of yeast transfer RNA with 77 bases was found out. It means from 1956 to around mid 60s, that is 1966, slowly and gradually, the development towards the advancement of the databases already started. From the first protein that was sequenced, that is insulin, to the tRNA, that is nucleic acid sequence of yeast tRNA with 77 bases was found out. During this period, three-dimensional structures of proteins was studied that are well known with the help of Protein Data Bank, which was developed as the first protein structure database with only 10 entries in 1972. Just imagine, only from 1956 to 1972, this is the journey of databases. This has now grown into large databases with over 10,000 databases. While the initial databases of protein sequences were maintained at the individual laboratories, as discussed just now, only because of the development of a consolidated formal database, which are known as Swiss protein sequence database, was initiated in 1986. Modern biological databases comprise not only data, but also sophisticated query facilities and bioinformatic data analysis tools. From 1956, 72, mid of 1960s, then 72, and then 1986. Slowly and gradually, the progress in the field of databases. That's why when such this progress is there, then they are considered as bioinformatic databases. When they are having query facilities and bioinformatic data analysis tools, then they are considered as bioinformatic databases. We have bioinformatic databases, mainly primary and secondary databases. They are divided into structures of databases, type of data, quality of data, maintainer status. Structure of database, that flat files in the form of flat files, relational databases, that is in the form of SQL, so, object-oriented databases, exchange publication technologies, that is FTP, HTML, COBRA, XML. Then types of data, that is in the form of nucleic acid data sequences, protein sequences, protein sequence pattern or motifs, macromolecular 3D structure, gene expression, and microarray, that is metabolic pathways also. Then quality of data, that is redundancy, checking data, human curation, updating data. Then maintainer status, large public institutions are involved. For instance, EMBL, NCBI, etc., etc. Academic group of scientists are also nowadays, as we just saw or discussed, specific universities or institutions are creating their databases and storing or managing themselves as well. Then commercial companies are also available nowadays just to create, just to manage, just to store or handle the various types of databases. Now, if we are having the huge amount of databases, this biological databases can be broadly classified into three databases. Sequence databases are applicable to both nucleic acid sequences and protein sequences. Structure databases are applicable to only protein structures, and pathway and databases, these are applicable only to pathways. When we are talking about the sequence databases as nucleic acid sequences and protein sequences. When we are talking about the nucleic acid sequences and protein sequences databases, this mainly represent the most widely used and some of the best established biological databases in the world. These databases serves as repositories for wet lab experiments or the outcome of the web lab, wet lab experiments, and the primary sources of experimental researches. That is in the form of experimental results, outcome of the primary source of experimental results. Major public data banks, which takes care of the DNA and protein sequences. These are GenBank in USA, EMBL, European Molecular Biology Laboratory in Europe, and DDBJ, that is DNA Data Bank in Japan. Some other sequence databases, that is GenBank, nucleotide database, EMBL, the EMBL nucleotide sequence database, DDBJ, DNA Data Bank of Japan, Ensemble, that is Ensemble database, SGD, Saccharomyces cerevisiae genome data database, is then DBEST, that is DBEST is the gene bank that contains sequences related with the expressed sequence tags, and TREMBL, European Bioinformatics Institute for translation of EMBL nucleotide sequence databases, PIR, that is the Protein Information Resources, SwissProt, Protein Sequence and Knowledge Database, UniProt, that is database is organized into 3 layers. On the basis of these 10 important bioinformatics databases, these are GenBank through GenBank, DDGB, and EMBL for nucleotide sequences, Ensemble, that is for human and mouse genome, PubMed, that is through NCBI, and literature references, then NR through NCBI for the protein sequences, SwissProt for the protein structures, Interpro, that is for the protein domains, OMIM, that is for the genetic diseases, enzymes through Expressi for the enzymes, DBP for the protein structures, CAC, that is genome for the metabolic pathways. These all are 10 important bioinformatics database according to the bioinformatics for dummies. Sequence databases having some technical definitions, data storage management, that is in the form of flat files, text files, human readable files are there. They have the relationship databases such as Oracle, Postgres, and the main purpose of this is object-oriented databases. First, fix your object, fix your motto, then only search with the respective databases of your desired information. These sequence formats, particularly in the form of BLAST prediction tools, FASTA, RAW, GCG, NBRF, PIR, MSF, or we can say that the standardized format for each and every database is available. Only in that format we have to work. We have to follow whatever are the instructions given by such database. For instance, Integrated Database Retrieval System at Genome Net, DBGET, an integrated data retrieval system. These are developed and maintained by the Institute of Chemical Research, that is Kyoto University, the Human Genome Center, that is University of Tokyo. These databases mainly covers nucleic acid sequences through GenBank and EMBL, protein sequences through SwissProt and PIR, 3D structures, PDB, Swift motifs, that is through PROSIT, Ligand reactions through ligand, literature search, that is LITDB, midline, PubMed, etc. You just, why we are uh, observing all these, or why we are going through all these, just observe this. Huge and huge tools are there, with they are forming the very dense network through which the information is retrieving through various databases. We have taken one example of DBGet search. This is the DebGet search. All databases, and these are the options which are available on the DebGet search. What we are looking for, that is search for either the all databases, either for ontology, pathway, disease, drug, chemical substances, chemical reactions, genome. Which is of our interest, we have to just click, and for either for humans, Homo sapiens, or for animals, any of the vertebrates or any invertebrates, or for microbes, or for viruses, plants, etc., etc. Put here and go. You will get the information related with your desired one on your laptop screen or computer screen. The another system is sequence retrieval system, that is SRS. First letter of all the three words sequence, that is S from sequence, R from retrieval, and S for system. Sequence retrieval system, SRS. This is the data retrieval tool developed by EBR, and it integrates 80 molecular biology databases. It is an open source software, can be installed locally, and has an associated scripting databases called ICRES. In which format now data is available, in which format we have to submit the data in the databases. This is the sequence database format, which is in the form of flat file. The example is SwissProt, that is protein database, flat file of this sequence database format. Having the accession number, taxonomy, reference is there, annotations, that is comments, and cross references. These are the 5 things which we have to submit when we are submitting our sequence to any of the suitable databases. What are they? For instance, we are submitting, for when we have submitted the sequence of Plasmodium citrullinalis, which is responsible for the degradation of petroplastics. That is, at that time, first we have obtained the accession number. Then that accession number we have to submit at the time of submission of sequence database. Then taxonomy. What is the taxonomy? For instance, we have selected here the human, right? And which erythropoietin precursor. This we have selected. This is protein we have selected. What is the taxonomy? That is Homo sapiens, is that belongs to human. What are the other? It is eukaryota, metazoa, chordata, and so on. Craniata, vertebrata, and so on. Now, which reference? That is sequence from where we have taken, that is where we have to submit, or on which basis we have developed our sequence. That is the reference of that sequence. That is sequence from NA midline. This number is this 85137899. PubMed is 383866. Then annotations, that is comments. What are the functions of erythropoietin precursor? Suppose this we have selected for its submission. What is the function of erythropoietin? Subcellular location, tissue specificity, and pharmaceutical availability is there if any product is related with the pharmaceuticals. Then cross references that are required, from where we get all these information. Here we get all these information from EMBL. All these we have to submit at the time of sequence submission of sequences in the sequence database in the form of prescribed format, just in the form of flat file. sequence database format, that is in the form of FASTA format. This is our sequence name. This is our raw format, and this is the FASTA format. Nucleotide sequence databases. This is database GenBank. This is respective URL. And what are the various features of these nucleotide sequences databases? This is NIH, that is National Institute of Health's Archival Genetic Sequence Database. EMBI's Archival Genetic Sequence Database. DDBJ, that is NIG's Archival Genetic Sequence Database. SGD is the repository for Baker's East Genome and Biological Data. EBI genomes, it provides access and statistics for the completed genomes. Ensemble, that is databases that maintains automatic annotation on selected eukaryotic genomes, particularly humans and mouse. Unigene, that is each unigene cluster contains sequences that represent a unique gene as well as related information. DBEST, that is division of GenBank, that contains expression tag sequence data. Likewise, protein sequence databases, these are the databases. SwissProt, TREMBL, these are the respective URLs of these databases. Excuse me. And what are the various features? For instance, SwissProt TRAMBL, description of the function of protein, its domain structure, post-translational modifications, etc. UniProt, central repository for PIR, SwissProt and TRAMBL. PIR, it strives to be comprehensive, well organized, accurate, and consistently annotated. PFARM, that is database of protein families defined as domains. And PROSIT, that is database of protein families and domains as well. Now, the first category is that is nucleotide databases, under which nucleotide databases along with protein databases. Now, the second category is structured databases. On the structured databases, immediately clicks in our mind, that is structure of the protein. Which type of structure, what knowledge of protein is required, which type of molecular interactions. These two are the key to understand protein functions and complex regulatory mechanisms underlying many biological processes. Structure databases, that is based on the structure of proteins. For that, which knowledge of protein structure is required, what molecular interactions are going on, in where such interactions are going on. In these two important facts, these two important factors, the secondary database is constructed or work. This mainly contains protein data bank, that is PDB, SCOOP, the structural classification of proteins, CAT, the class architecture topology, homologous superfamilies, and NDB, that is nucleic acid databases. These are PDB, respective URL is there, and features are protein structure repository that provides tool for analyzing these structures. SCOOP, classification of protein 3D structures in a hierarchical scheme of structural classes. CAT, that is hierarchical classification of protein domain structure. NDB, that is database housing nucleic acid structural information. The third category is pathway databases. Development of metabolic pathways, that is metabolic databases, which are derived from the comparative study of metabolic pathways, will cater the industrial needs in more efficient manner to further the growth of systems biotechnology. These, for the proper growth of systems biotechnology, some examples of pathway the databases are CAG, Brenda, Biosys. CAG is the the Kyoto Encyclopedia of Genes and Genomes. Brenda is the main collection of enzymes functional data, and Biosys, that is collection, is a compilation of pathway and genome information of different organisms. These are the pathways of these are the databases of the uh, pathway databases. These are the pathway databases, URL, and the specific features. CAG, protein structure repository, that provides tools for analyzing these structures. Number one. Biosys, that is classification of protein 3D structures in a hierarchical scheme of structured classes. Brenda, that is hierarchical classification of protein domain structure. EMP, that is database of enzyme and metabolic pathways, public server. And Bright, biomolecular relations in information transmission and expression. These are the pathways of the three different categories, three different classes, that is nucleotide pathways, structured pathways, and pathways for the various pathway databases for to study the various pathways for the metabolic purposes, metabolic pathways. Organisms division is there on the basis of the name of their organism and which type of tool they are having. For instance, BCT, BACT, for the bacterial, tool available is DDBJ and GenBank. Whatever the information you want related with the bacteria, all are available in this DDBJ and GenBank, that is through BACT. Fung for the fungal, EMBL. Hum for the Homo sapiens, DDBJ and EMBL. INV for the invertebrate, that is for all type of databases are applicable here. MAM, other mammalian, and all databases are available here. ORG for the organelles, cell organelles, that is applicable here are AMBL. PHG, that is the phage. All databases are available. PLN, that is plant. All databases are available. PRI, that is primate. Also, we can consult the HUM for searching of the appropriate results, appropriate queries, we can say. Uh, these all are available in all the databases, but not same data in all. All databases are having different data. Although the query is same, but the result of the data from the various databases are different. PRO, that is for prokaryotic, EMBL, available on EMBL. ROD, rodent, that is all. SYN, that is synthetic proteins and synthetic enzymes, available on all. VRL for the viral, available on all. VRT, that is other vertebrate, that all informations are available on other all databases. Nowadays, slowly and gradually, the advancement towards these databases are also developed. Likewise, when we are just subcategorizing the subjects in the broader areas or the broader fields, for instance, biotechnology, one is genetic engineering, another one is molecular biology, third one is biochemistry, fourth one is immunotechnology, stem cell biology, etc., etc. In the same manner, databases are now more and more in the more and more manner they are refining. We are nowadays we are having the databases that are exclusively human related biological databases. Each and every aspect related with the human. For instance, names are 1000 genomes present here. This is the URL, and a deep catalog of human genetic variations. Category is DNA. AFND, AFND, that is allele frequency net database. DBSNP, that is database of single nucleotide polymorphisms. DEG, that is database of essential genes. EGA, that is European Genome Phenome archive, es- es- ensemble, that is ensemble genome browser, UGs, that is genomic information for eukaryotic organisms. Likewise, so many, if you want to deal with the transcriptomes, if you want to deal with the long non-nucleotide RNAs or miRNAs or nucleotide sequences or PDBs, that is protein sequences of the smallest or the largest part or the fragment of our human, that all are available in one way or the other in all these databases. But as soon as the more advancements are there in the field of databases, the latest discovery in the field of databases are best match, new relevant search for PubMed. Everybody knows PubMed is a free search engine for biomedical literature, accessed by millions of users from around the world each day. Whatever we are discussing or whatever we have discussed, all we have just gathered from or searched from the PubMed. But with this rapid growth of biomedical literature, about 2 articles are added every minute on average. Finding and retrieving the most relevant papers for a given queries is increasingly challenging. Hence, best match, a new relevant search algorithm for PubMed that leverages the intelligence of our users and cutting edge machine learning technology as an alternative to the traditional date sort order. This best match algorithm is trained with past user searches with dozens of relevance ranking signals, that is vectors, the most important being the past usage of an article, either what is the publication date, relevance score, and type of article that are required whenever we are using the best match. This new algorithm that mainly demonstrates the state-of-the-art retrieval performance in benchmarking experiments, as well as an improved user sequence, that is in improved user experiments in real world, estimating over 20% increase in user click-through rates. Just over 20% increase in user click-through rate. This was developed in June 2017. Since its development, a significant increase, that is 60% in PubMed searches with relevant sort order, has been done so far. It now assists millions of PubMed searches each week. Just to increase the awareness and transparency of this new relevance sort option for PubMed users, enabling them to retrieve information more effectively in a very short span of time. This is the overall architecture of the new relevance search algorithm in PubMed, which mainly consists of two stages. The first is so that is BM25, which is considered as classic term weighting algorithm. The top 500 documents, they are resulted and then they re-ranked with the help of Lambda Mart. Here, the document is there. Here, our query sequence. This is mainly working on offline training process. Featured depends upon our query and present on the document. All these are just mixing here or aligning here in the lambda mart. Then with the help of the search index, whatever is the new query, that is just indexing or that is just uh, in entertaining on the basis of BM25, then this is the search which this tool is doing with the help of top 500 documents. Then they again rank that on the basis of the results produced by this L2R algorithm. Here we are getting the results of lambda mart, and from here, that is BM25, top 500 documents are then re-ranked, and whatever the results with the help of this model we are getting, this is the results of the final one. On the basis of whatever the results we are getting, then what happened? They are just showing the actual ranks of that 500 results. These features mainly designed on the basis of machine learning based ranking model, which is mainly learned offline using relevance ranked training data together with a set of features extracted from the query and the documents, or both, either from the query or from the document or from both. This is mainly designated as D for the document, ID for the inverse document frequency, L2R for the learning tool rank, then Q for the query, and query, then QD for the query document, and TIAB for the title and abstract. In this manner, we are getting the complete information along with their relevant store within few seconds of time. That is known as best match. Hence, all these which we have discussed with the help of some references which we have used for the preparation of this presentation. Now we are summarizing whatever we discussed in the first session of the fourth workshop. That is, we are knowing very well that the biology is developing, or biological sciences are tremendously growing day by day with, with increasing turns into a data-rich science. That is the need for storing and communicating large data sets. Some obvious examples we have already discussed. That is in the form of nucleotide sequences, protein sequences, and the 3D structural data that are produced by X-ray crystallography and NMR. Nowadays, we cannot imagine our experiments without the presence of biological databases that are our assisting scientists to understand and explain a host of biological phenomena from the structure of biomolecules and their interactions, or to study the whole metabolism of organisms, or to understanding the evolution of species. Whatever the thing which we want to study, which we want to analyze, without these biological databases, even we cannot think. This knowledge helps facilitate the fight against even the disease we want to discuss. Suppose we are working on T2DR, that is type 2 diabetic retinopathy. We want just the H3 of T2DR of BDNF gene we want to study. In which manner we have to proceed? That is with the help of biological databases. Or in the development of medications, that is prognosis, diagnosis, and therapeutics. These databases are useful. And in the discovery, basic relationship amongst species in the historic of life, that is phylogenetic relationships. Evolution, which one is evolved from which one. All these we can study with the help of biological, or we can design or frame out with the help of biological databases. Thank you so much for the patient hearing.